Good afternoon, and welcome to our Biomedical Informatics and Data Science Grand Rounds lecture series. My name is Paul Naji. I serve as the program director for our online master's training program here at Johns Hopkins in our School of Medicine. And really, it's a pleasure here to have Tom coming to talk to us today. I hope that we've talked a lot at Johns Hopkins about our precision medicine platform, how we're trying to help our physicians turn their clinic into a laboratory and build a learning health system. And so what's really important is understanding how other health systems are doing this, doing very similar initiatives, and how really this is a trend throughout the entire field and so really we're excited to have Tom here talk to us about how he's doing that. And so Tom, Dr. Campion leads the Weill Cornell Medicine's efforts to support clinical and translational investigators with electronic patient data, especially through the secondary use of electronic health record data. He is an associate professor of research in population health sciences in the Division of Health Informatics, and as the Chief Research Informatics Officer in the Office of the Senior Associate Dean for Clinical Research and the Information Technologies and Services Department, as well as the Director of Biomedical Informatics in the Clinical and Translational Service Center, CTSC. He leads the architecture for research computing in health, as well, which matches scientists with tools and services for obtaining electronic patient data. He has served as a co-investigator in multiple funded research initiatives, including the NIH's Recover and our very own N3C, as well as ACT and All of Us research program, as well as PCORI's Insight Clinical Research Network. And nationally, he leads efforts to advance the secondary use of EHR data through the NIH CTSA Consortium, Clinical Research Forum, IT Roundtable, as well as the Association of American Medical Colleges, a group on information resources. And so, Tom, thank you so much for joining us today. We just had an incredibly eventful AMIA meeting, and we're all excited. Our students are back, and we're really eager to talk about informatics and what we've learned and how, as a field, we're really at such a great time in how we can help our clinicians understand their patients and help them discover things about them. So it's really exciting to have you here. If you want to share your screen, we really appreciate your talk. And so, as an audience, please make sure to ask questions. Harold and I will be asking questions at the end. Please add them into the Q&A section or through the chat, and we'll make sure to have them asked at the end of the lecture. And with that, Tom, please take it away.
Terrific. Thank you, Paul. Thank you, Harold, and Stacy for hosting me. It's a pleasure to be here virtually with you all. And to get a chance to share a bit of our approach at Weill Cornell Medicine. Just to reiterate, I have no disclosures to make, and the goal of today's talk is to really focus in and help everyone understand how to go about doing this matching of tools and services with investigators so that they can do science. So a little bit about how this will be structured. First, I will describe the problem that we've conceptualized with supporting the biomedical research enterprise. Then I'll go a little bit into some critical organizational background. And then what I really want to spend most of our time on is going through both organizationally how we support research computing at Weill Cornell, and also tactically how we do it through something called Architecture for Research Computing in Health, or ARCH. And if you have any questions as we go along, I'll try to keep an eye on the chat, but maybe we can get to those in total at the end. So as you very likely know, getting data out of electronic health record or EHR systems to do any type of analytics, but research in particular, is hard. And it's hard for a number of reasons. First and foremost, EHR systems are not designed for research. These are designed for individual patient care transactions, really modeled after the manila folders of yesteryear, one for each patient, filled with single sheets of paper, a progress note here, a lab report there. that were, really intended to present all of the information to a particular clinician when that clinician, say, is interacting with a patient in an exam room. These are individual patient care transactional systems. And as we digitized all of this, especially in the US, we really kind of designed them first and foremost for billing, probably secondarily for patient care, and in a distant third place for analytics and research. So we're constantly having to take this transactional data and transform it into an analytical data set. And we'll talk more about that as we go along. Additionally, there's a variety of different tools to choose from in clinical research. And when you look at clinical care, one can argue that Epic has solved this problem, that Epic is the one system to rule them all. I know there's lots of holes in that argument, but by and large, you look at things like market share, Epic is the go-to system, particularly in academic medical centers for clinical care. But when it comes to clinical research, there is no single system to rule them all. There is no Epic for clinical research. Instead, we have a variety of different tools and services, and it takes a bit of knowing and doing to navigate all of those options. So with all of those different tools and services, they all have their own strengths and limitations. And at the foundation of all of this in the EHR, the EHR data really has a series of strengths and limitations as well. Investigators come to our group all the time and ask, can I do study X with data from the EHR? And I give the same unsatisfying answer every time of, well, it depends. And it depends on a variety of factors. Do the data exist in the EHR at all? We got started with the EHR system here in the 2000s. If you want data from 1985, we don't have that data. But once we start getting into the data, taking a look at whether elements are structured, things like CPT codes, quantitative lab results that lend themselves more easily to automated extraction versus, say, unstructured notes that may require things like natural language processing to really derive clinical concepts from them. These are all some of the things that we need to understand as we evaluate what the literature calls fit for purpose. So along with those strengths and limitations is also the need to obtain regulatory approval. Patient privacy is of paramount importance. And it's really hard to navigate all of the different approvals that are necessary, whether it's from your local institutional review board, or maybe you need a contract in order to share data or receive data with another institution. These things are really hard to follow, and the majority of clinical and translational scientists are not up to speed on all of this, and perhaps why should they be? This is really pretty nuanced, but we're still talking about the most sensitive data about people, and it is absolutely critical that as stewards of this data, that we respect patient privacy. So kind of taken together, this is a very complex socio-technical problem. And when we look across the biomedical informatics literature, in particular in kind of the clinical research informatics and translational bioinformatics sub areas, it's pretty clear that optimal approaches are unknown. What we're trying to do in the field right now, and at Cornell through our group, is really test hypotheses to see what works, to enable the research enterprise to do science using EHR data, and what doesn't, so that we can constantly adjust our approaches and accelerate time to science. So there's really three things that kind of broadly the field of research informatics, in particular, our group at Cornell called Research Informatics helps investigators do, and that's to get EHR data, to collect novel measures that may not exist in the EHR, and integrate data from disparate sources. as we kind of go through things, I'll illustrate what this looks like. A bit of background to get started. As you may know, Cornell University is located in Ithaca, New York, which is in upstate New York, and is an Ivy League institution. And it has a variety of different schools and colleges. One of them is the medical school, which is called Weill Cornell Medical College, and is located not in Ithaca, but in New York City, specifically at Manhattan on the Upper East Side. So we're about 4 hours away from the mothership. Kind of function semi-autonomously. A lot of things like the grants and contract system, the IRB system, that's all handled here at the medical college, separate from the main university. Lots of times when these schools are in closer proximity, they may share some of those functions. But at any rate, Weill Cornell Medical College is the biomedical research, education, and care unit of Cornell University. And it employs about 1,600 physicians who are all faculty members of the medical college. They practice in outpatient settings throughout the greater New York City metropolitan area, doing business as Weill Cornell Medicine. It was a branding change a couple of years back. Also and previously known as the Weill Cornell Physician Organization, but 1,600 docs, all practicing, all faculty members of Cornell. And since about the year 2000, they have used the Epic electronic health record system in all of those outpatient practices. Now, one thing that's important to understand here is that Weill Cornell Medicine and Cornell University does not own a hospital, just employs physicians and owns the outpatient practices. When patients of Weill Cornell physicians get very sick, our physicians have admitting privileges to a longtime clinical partner, that being the New York Presbyterian Hospital System, which has about a dozen facilities throughout the New York City area. And for a very long time, it ran the Allscripts EHR system on the inpatient and emergency side. There are a series of automated interfaces between Epic and Allscripts, between Cornell and New York Presbyterian or NYP as we call it. Enterprise patient index, a lot of automated transmission of clinical data, but it was still two different EHR systems, one for the outpatient arena and one for the hospital setting. So all of this that I'm talking about with respect to Cornell, it also applies to our colleagues across town at Columbia University, which similarly is a university that has a medical school, and the medical school then has a physician organization doing business as Columbia Doctors. And for a long time, they ran Allscripts in the outpatient setting. And just like Cornell, Columbia does not own a hospital, instead has their physicians have admitting privileges to New York Presbyterian. And New York Presbyterian that had a separate Allscripts implementation for the Columbia portion, kind of separating Cornell and Columbia. So this made for a great deal of challenges. And over the past 5 years or so, the three institutions who are, very much aligned on a lot of different things, but also, competitors when it comes to outpatient activities, their research enterprises, medical school admissions, and so on. The three institutions came together and said, you know what, let's take Cornell's Epic implementation and share it across the three institutions. This has been a great thing for clinical care. About 12 million patients data across dozens of locations, throughout the tri-state area are now on one Epic instance. That's really great for patient care. It gets particularly challenging for analytics and research, and I'll try to showcase a little bit of that today. So, given that organizational background, just want to kind of emphasize that the work that I'm describing here today is what we do at Weill Cornell Medicine to enable scientists, and a little bit about how we're organized to do that. It all starts through our IT department, and our IT department, which is called Information Technologies and Services, or ITS, provides everything from the network jack on up. So, email, servers, information security, kind of these traditional IT sorts of activities. They're all kind of represented in that gray box of that foundation of IT infrastructure. And on top of that foundation, we have 3 specific divisions that support the needs of scientists across the spectrum of activities from conduct to administration. So on the conduct side, kind of starting there, our scientific computing group really delivers high performance computing, the types of storage and compute that you need for, say, genomic sequencing and analysis. Just to put things in perspective, one whole genome sequence is about 1 terabyte of data. That's for one patient. When we look inside of Epic, we've got data for millions of patients. It's about 50 terabytes of data. That's for 12 million patients or so. If we want to sequence everyone who comes through the door, we're going to need a lot of terabytes of storage. So that's why there's a particular group that focuses on that. Switching gears to the other side of the spectrum, the administrative side, our research administrative computing team, they do kind of all the compliance and planning activities, the grants, the contracts, the IRB protocols, what you can and cannot do, who's enrolled in particular studies, and how do you go about paying for it. And then in the middle is our research informatics group. We bring together the activities of these 2 scientific units, and also the clinical enterprise, which is not shown here, but I'll try to illustrate this as we go ahead. So the confluence of all of these activities from these different organizational units is a suite of tools and services that we call Architecture for Research Computing and Health, or ARCH. And as you can kind of see here, there's a variety of functions that span a spectrum of study, from retrospective to prospective. Another way of thinking of it is from the study of populations to individuals. Some activities that are cross-cutting, things like consent that may pertain to all types of study design. But we really try to bring all of these things together because it is exceptionally tough to do on your own. I should also point out that early in my career, I learned that to succeed in academic medicine, you need lots of lousy acronyms. ARCH is but one of those. There will be more. Consider yourself warned. But kind of focusing in on the types of activities that our research informatics team does, it's really the functions represented by these red boxes. I'll illustrate as we go along how these different functions manifest in one or more systems. But I remind our staff all the time that science doesn't happen in the systems represented by these red boxes. Science happens in a statistical software package like SAS or Stata or R, because it's using statistical software on the data that we provide that investigators can, say, conduct epidemiological analyses, develop new predictive models, really come up with the discoveries that help drive biomedical science. And there's a citation here from a JAMA article that I'm quite fond of that says, in academic medical centers, we're great at breakthrough, these discoveries. We're lousy at follow through, that we don't take these models and use them in practice. But the electronic health record provides a platform for doing just that, where we can take our models and make changes in the system to drive changes in human behavior. This could be through best practice advisories. It could be through order sets. It could be through visualizations, a variety of different types of things. And as we put those interventions in place, we can then get data back out of the EHR system and enable us to understand, okay, we made a change in clinical practice. Did we make things better, worse, or have no effect? This data-driven feedback loop is what policymakers call the learning health system, and that's what we're here to enable. So, I mentioned that the different functions manifest in a variety of different applications. And that's what you can see here. The purpose of this slide is not for you to look at it and understand, oh, okay, now I know what to do. The purpose of this slide is to illustrate that there's an alphabet soup of acronyms, of product names, that there's so many different tools and services to choose from that it's really hard to navigate this. And that's where our research informatics team comes into play to help navigate investigators to the right tools and services with respect to things like study design, data sources, cost, and other considerations that we'll kind of step through today. So a little bit about how we're organized. Within our research informatics division, we have 3 sections. One that handles the data and software engineering that is crudely reduced to that cartoon at the top. SUPER is the name of our data processing environment. It stands for Secondary Use of Patients' Electronic Records. I told you there would be bad acronyms, and we'll kind of step into that in just a moment. And then we have 2 different groups that really focus on engaging with investigators. Our data science services is really focused on secondary use of EHR data, and our clinical and translational services section really focuses on systems that drive particular scientific workflows, really kind of individual transactions. These are things like REDCap, Open Specimen, which we use for biobanking, a variety of other applications. As we kind of discussed a few moments ago, we have a kind of a bifurcated organizational existence, and we bring in a variety of data from both Weill Cornell Medicine and from New York Presbyterian. This figure is a bit out of date, but it gives a sense for the number of systems from which we draw data. And then once we have the data, what we're really focused on is data delivery. That's the business that we're in. And what investigators want and need are research-ready data sets, but the data that come out of these source systems, whether they're clinical or research systems, they're pretty raw. And we go through a series of steps that kind of go up this stack for us to be able to deliver that research-ready data. And fortunately, we don't have to do some things like our own information security, the networking, the servers. Our traditional IT colleagues provide those services for us. But we've estimated using ServiceNow, which is our service management platform in our IT department, that more than half of our group's time goes to really kind of these activities under the surface, all the data integration, cleaning, and quality assurance that we need to do to be able to put it into the systems that we took a look at on the previous slide. This is a great deal of work, and I think kind of where a lot of effort across research informatics goes today. But we really want to be at the top of this stack. I want to get out of doing so many of these things further down in the stack, as I'm always kind of taken by this concept from clinical medicine of performing at the top of your license. I think the role of informatics is to perform at the top of this, of navigating these research-ready data sets, and trying to spend less time on the things further down in the stack that other professionals may be able to accomplish, but we as informaticians really deliver a lot of today. So kind of coming back to this, I really kind of want to now walk through a lot of what these different tools and services are. And kind of the bread and butter is electronic health record reporting, of getting, kind of rows and columns of data. And I mentioned that historically we've had multiple EHR systems managed by our two institutions, Weill Cornell and NYP, and also sharing that with Columbia University Irving Medical Center. So there was always a big kind of question of how do we get data from the EHR systems, and through a series of legal agreements and cooperation across the three institutions, something called the Tripartite Request Assessment Committee, or TRAC, came about. And this is really a single front door for requesting data from the EHR. And what that really looks like is transforming that raw data into something that's research ready. It's kind of what I want to show in these next few slides. So this may look familiar to some of you. What we're looking at here is indeed rows and columns of data. But I don't think it's the type of rows and columns of data that investigators typically want to use in something like SAS. Instead, this is the raw data in the Epic Clarity reporting database. I've obfuscated a couple of fields here, but you see, different columns, patient number and encounter number, as a patient could have, one or more encounters. And then something called DXID. And you kind of look at DXID and been around healthcare and I think DX, yeah, it's probably, diagnosis ID. But looking at these numbers don't make any sense to humans. These numbers might make sense to machines. They're machine readable. They're not human readable. And it's only when you join in data from another source, like let's say a lookup table of ICD-9 codes, that the data start taking on a little bit more human readability. Just off the top of my head, having been around healthcare a little bit, I know that anything in the 250 range of ICD-9 codes, that pertains to diabetes. So, kind of thinking about like, what does an investigator want in a rows and columns level data set? It could be that they want these individual diagnosis codes, but let's just consider, for example, that they just want dichotomous variables of yes or no. Does this patient have a particular comorbidity? Because what investigators probably want is not this type of rows and columns level data out of Epic, but really something like this, where each row represents one patient. And then we have a series of dichotomous variables, yes or no, does this patient have diabetes? We could have set a rule based on, do they have an ICD-9 code in that 250 range? And then repeat this process for other comorbidities, maybe using diagnosis codes as well, or we could use, combinations of lab results, and then to repeat this for other elements from the chart too, things like medications, maybe social history. Now, I'm not saying that, dichotomous variables are what people only ever want to study. This is just one example. But kind of thinking of, all right, how do we deliver this research-ready data set so that someone could perform this type of analysis to do all of that data cleaning? That's kind of what we're aiming to do. Now, to deliver one of these takes a lot of back and forth between a requester and a database analyst. And the database analyst, wants to know, okay, you want all of the blood glucose values. Do you want the blood glucose value that appears in the upper left-hand corner or the lower right-hand corner of the screen? Because that kind of determines, where they'll go and extract it from. And this back and forth process can take anywhere from days to weeks to months to complete. This is typically, really something that's useful when it's time to perform analysis of data. But a lot of the time, investigators aren't quite ready to analyze data. They might be at a more formative stage of the research process, where they're doing things that are often called preparatory to research, of really trying to understand how many patients are there with X, Y, and Z. And for a number of years, we've used a platform called I2B2. It stands for Informatics for Integrating Biology in the Bedside. It came out of Harvard Medical School and the Mass General Brigham group. And with I2B2, basically you take all of your EHR data and you transform it into a particular format that I2B2 supports. And then investigators can navigate all of the data in a hierarchical form, as you can see here, and they can click and drag and run queries. And in a matter of seconds, using self-service tools, they can find out, for example, how many patients ever had an ICD-9 code in that 250 range. And that's what we can see here. Now investigators can add in other elements too, laboratory results, say, and the patient had, 2 hemoglobin A1Cs over 6.5, maybe some medications, and the patient is on insulin, that sort of thing. And then and that all investigators can use this without IRB approval, as we've de-identified the data, and they're only getting counts of patients, not actual data. But if they then subsequently obtain IRB approval, saying, yes, I need to study, these 1,000 patients who have this particular condition, and I need to obtain protected health information for these patients. So long as that's approved in an IRB protocol, we can move forward with that. And we do probably 60 to 100 of those per year. So the advantage of I2B2 is it puts data from our institution at investigators' fingertips. But as you know, healthcare in America is fragmented. And in New York City, it is particularly fragmented. As we often see that a patient will have a primary care physician at Weill Cornell, but their endocrinologist is at NYU, and their cardiologist is at Mount Sinai. And then they slipped and they hit their head, and they ended up in the emergency department at Montefiore in the Bronx. So to really try to understand the full continuum of care, you need to bring together the data from all of those different institutions. This is also very critical to the study of rare diseases. Through PCORNet, the Patient-Centered Outcomes Research Network, funded by the Patient-Centered Outcomes Research Institute, or PCORI, Weill Cornell is the coordinating center for the Insight Network, which brings together all of the EHR data from all of the academic medical centers in New York City. This has been going on for a number of years now. Renu Kaushal, who's the chair of Population Health Sciences and Senior Associate Dean for Clinical Research at Weill Cornell, is the principal investigator. This has been a remarkable resource for research. Remarkable to bring together all of these clinical competitors and get them to share their individual row-level EHR data, because everyone knows that doing this is the right thing for science. Just a remarkable partnership. In addition to what we do with Insight, we also have a number of other multi-institutional data sharing initiatives, one of which some of you may know, with a company called Trinetics. And with Trinetics, Trinetics recognized a long time ago that providing counts of patients meeting criteria from electronic health record systems would be valuable not only to investigators in academic medical centers, but also to biopharmaceutical companies. And this has really been a great fit for our institutional risk management policy. We're a pretty conservative institution when it comes to patient privacy, that we don't just want to give data to commercial partners. But with Trinetics, we saw that pharma would get counts only. They would never actually get real patient data. And that this really would help us with encouraging sponsor-initiated trials. This figure is a little bit old, but you can get a sense here of in the past 4 or 5 years that we've been doing this. We've gotten, well, once I update this figure, it's much closer to about 350 or 400 clinical trial opportunities that come in. And every year we go and ask our clinical trials office, do you want to keep doing this? And they say, yes, we're getting trial opportunities that we wouldn't ordinarily have gotten. So this is a great way to continue bringing cutting edge therapeutics to patients at Weill Cornell while still respecting patient privacy. Much as Trinetics helps with growing sponsor-initiated trials, through the NIH, something called the ACT network, which stands for Accruals to Clinical Trials. We've been involved there to help drive investigator-initiated trials. This leverages the I2B2 infrastructure in place at nearly all CTSA hubs and connects everyone together. And almost all CTSA hubs are now participating. And I think the participation, there's a fair bit of talk at AMIA. This is now evolving into something called the ENACT network. I think the N stands for next generation. So more to come with the ACT network. But over time in this figure, we've also added in this big data analytics set of activities with the Observational Health Data Science and Informatics, or Odyssey community. And we really kind of started honing in on Odyssey for a number of reasons, but especially as we were getting more computationally sophisticated investigators at our institution saying, hey, you know what, I want all of the EHR data to run unsupervised machine learning algorithms. We were kind of thinking, oh wow, I mean, all the Clarity data in Epic, it's pretty hard to understand. It's proprietary. There's a saying, if you've seen one Epic implementation, you've seen one Epic implementation. And just thinking there's so much to navigate here. It's really hard. Do you really want to have to do that? And a number of years ago, this headline appeared in the New York Times about janitor work as being absolutely critical to supporting big data. We kind of looked at it and we thought, well, can we do this at scale? And what if we were to do this using something like Odyssey, and particularly the OMOP Common Data Model, the Observational Medical Outcomes Partnership, which isn't quite all of the data, but it's kind of everything that you actually need, with 20 standardized tables. The beautiful thing about OMOP is if I implement OMOP correctly at my institution and you implement it correctly at your institution, we can run the same queries, and perform the same analyses, and generate these aggregate numbers, and we can compare those and do science. And we never have to move data across organizational boundaries, which means we never have to involve lawyers, which can really help accelerate time to science. Some of you may be familiar with OMOP, but this is this figure is a representation of the data model. On the left-hand side are kind of common clinical concepts we often encounter. Things like a person who has a visit occurrence, a procedure occurrence, a drug exposure, and so on. And what's critical about OMOP is that all of these clinical data elements are mapped to standardized vocabularies, things like SNOMED, LOINC, and others. I think this is just a wonderful example of how we've been able to take the past 50 years of medical informatics scholarship, both in kind of theory and practice, and bring it together so that we can have data that's encoded in reference terminologies that have really gotten better since the Meaningful Use Program went into effect in about 2010. And having all of this clinical data and encoded in these reference terminologies, then available in a data model on the left-hand side of the screen, that really helps people, be a quick study. This is remarkable and has really helped guide a lot of our internal data science initiatives as we seek to grow self-service. OMOP and the Odyssey tools have become a major part of our multi-institutional data sharing initiatives as well, including the All of Us research program. For those of you who don't know, the NIH started the All of Us research program a number of years ago to help enroll 1 million patients and 1 million people in the United States to donate their electronic health record data, plus genomic data, and a variety of other elements from things like surveys and Fitbits, to enable a massive cohort study. I kind of look at this as the Framingham Heart Study, meets 23andMe. The great thing, and this was outstanding to see at the AMIA symposium earlier this week, is all of the data are available today to investigators. There's a trend these days not to send data to investigators, but to send investigators to data. And that's what the secure web-based researcher workbench enables investigators to do. You can see a couple of different features here. And by going through this approval process, investigators can get access to EHR data, electronic patient data, in OMOP format, and then use common tools like Jupyter Notebooks to be able to generate things like sunburst plots, write your own queries, and really just do outstanding data science work at scale for patients who have consented. This OMOP data has also been critical for Weill Cornell to participate in the National COVID Cohort Collaborative, or N3C, which many of you are familiar with. Briefly, and these stats on the left-hand side are a few months out of date, but early in the pandemic, this was just an outstanding example of the informatics community and the NIH doing what is right and helping to bring together EHR data to address the most compelling public health need of our time. And again, it's the sort of thing where you bring researchers to the data. This is a big centralized resource, and all of the data is available for query in the OMOP Common Data Model format. You can see a SQL query shown here, which is just a great way to enable data science. So another element of kind of big data analytics to share is some of our activities in natural language processing, or NLP. Most of what I've discussed today so far has been about structured data, but what about unstructured data? Things like physician notes, where that's might be where the true state of a patient may be described there more so than in, say, some structured terms. Over time, our group has targeted NLP activities on particular use cases guided by our clinical partners. You can see a variety of things here. Things regarding mental health, like depression. We've more recently done some work in suicide ideation, but also extracting things out of pathology reports, ejection fraction, a bunch of elements from bone marrow biopsy reports. But one thing that I want to focus in on here today is some of the work in health equity we've done around race and ethnicity. So as you know, Black and Hispanic patients are traditionally underrepresented in biomedical research, but it's really hard to try to recruit patients from these communities because race and ethnicity data in EHRs are often incomplete. And when we looked at the structured data in our EHR system, we found that most of these values were declined or null and just not specified. And there's no real way of knowing, did someone actually state that they declined to share this? Or was it, a quick maneuver from, say, a registration desk clerk who just wanted to get someone into the institution, clicking boxes in the EHR system quickly? We don't know. But we hypothesized that if we were to take a look at unstructured data in the EHR from notes, that there might be a few clues that could help us kind of fill in the blanks. So here we're looking at the history of present illness from a progress note, and kind of see here that it starts off 48-year-old AA female, presumably where AA means African American. we kind of started thinking, well, could we use that to kind of fill in these gaps? And we were able to demonstrate in a paper from a few years back that we could identify 26% more Black patients and 20% more Hispanic patients using this NLP approach. And this was all geared towards clinical trial enrollment. And we're also able to note health disparities among these patients. I'll make sure to share a link to this article in the slides that get distributed. But one point that I want to kind of bring back here is that all of these elements that we've been discussing and are, efforts for us to get structured concepts from unstructured notes. And it's not just that we do that kind of in a one-off fashion. It's that we then take that, we make it, we put it back into OMOP, and so that our investigators can then use that. And then, OMOP then undergirds what we do with a lot of multi-institutional data sharing initiatives, including with Trinetics and N3C. So it's kind of constantly enriching the data that are available in self-service tools at our institution. So I've been talking a lot about activities kind of on the retrospective side of the spectrum. I kind of turn a little bit to the prospective side of the spectrum too, because sometimes what investigators need are kind of small data sets, and that an electronic data capture tool like REDCap is the way to go. Over time, we've also been able to automate a lot of the data through something that we call Super REDCap. Which is just kind of internal branding of some of the features, the dynamic data pull and clinical data pull features that Vanderbilt, the makers of REDCap, put in place. And I just kind of want to briefly touch on what these things are. So for those of you who aren't familiar with REDCap, it's kind of a build your own case report form and survey platform, but really optimized for research, kind of like a SurveyMonkey or Qualtrics, but for research. If you're an academic medical center and you're meeting certain security standards, you can indeed store protected health information in it. And the folks at Vanderbilt that created this, they saw that all too often investigators had REDCap open on one screen and Epic open on the other. And they developed a feature that we've branded as Super REDCap that says, all right, if you enter in a medical record number, the system will recognize that and say, we'll go and get the data out of your remote system, in this case, an EHR system, and bring it back for you. So in this particular example, we're talking about kind of first name, last name, and date of birth for a test patient. The investigator would look at this, say, yep, looks right, press save. We've had this in place for a number of years now, and investigators have let us know that it helps save time and reduce data entry error. And that's a massive boon to the research enterprise. In addition to kind of doing those kind of one-off elements, it can also support temporal variables. So based on the combination of a medical record number from the previous screen and a date, it'll go and retrieve, say, all of the platelet values. So the investigator can select that platelet as opposed to this platelet. As so much of the time, investigators are tasked with really kind of annotating data and kind of transforming it by hand. Where doing so in an automated fashion may not be most expeditious. We were also one of, I think, the first 8 sites globally to implement REDCap on FHIR integrated inside of Epic. And this really helps further bring together clinical and research workflows. And our investigators find this to be particularly beneficial. But a number of years ago, we recognized that investigators would have data in a REDCap project and an Excel spreadsheet that they'd be able to export. And they would have another spreadsheet that they got from the Epic reporting team describing outpatient behavior, another one from the Allscripts team describing inpatient visit details, and then another from, say, the perioperative system, and then a standalone spreadsheet about their biobank, and all of a sudden they were swimming in spreadsheets and they're saying there's got to be a better way. So we devised an approach that we call research data repositories, whereby we bring together all of the data for a particular group. In this case, we're kind of using an example of emergency medicine, and then we regularly refresh all of the data from all of those disparate systems. We then deploy 3 different tools that support 3 different workflows. The I2B2 and Super REDCap activities we kind of went over, but the big one, and where we spend the majority of our time, is that final workflow of analysis. And that's where we focus on developing those rows and columns level data sets. As we've gone through these RDRs, as we call them, we've recognized that it's nearly impossible for us to create one database, one pluripotent database. But instead, if we focus on kind of 2 data marts to support particular research questions or families of research questions, that we can go far. So for example, for our digestive care group, we had study one focused on colorectal cancer surveillance, and study 2 focused on cirrhosis. Might be some overlap in patient population there, but it's really different rows and columns, different variables of interest. And we've had great success with this, of being able to engage maybe 20 or so different investigator groups, as shown here. And this has really led to a great deal of development of these resources, of learning about our data, not just for informaticians, but also for our clinical partners, so they can understand the strengths and limitations of EHR data for research. And over time, we've created, a number of data marts that we now publicize in something called the Data Catalog, which is really there to help encourage, groups to come together to use, these data sets that have already been made research ready, so that we don't have to go and, reinvent the wheel. And of course, as we take a look back over the past few years, the pandemic has been a major part of everything that we have done. And early in the pandemic, we saw that we had an unprecedented number of requests for data, and that our typical approach of really doing a lot of investigator handholding would not work. And instead, what we needed to do was to provide self-service, but to really kind of use a lot of our existing approaches, including bringing together all of the data from systems of interest. And then instead of working with investigators to kind of define the elements of interest, we just drew from our experience for kind of more common things, some of the domains of which are here, to support a variety of different use cases, and then kind of start wrapping our existing compliance and engagement processes around it, so that we could serve investigators. I ended up publishing a paper in IJMI describing some of this, and how some of the automated data that we assembled in an automated fashion compared to some of the manual data that a team of medical students had abstracted in REDCap project very early on, and kind of tacking on biospecimens and a whole request process, as described on some of our internal websites. So as we kind of bring things back here and reflect on all of this, just a couple points to make. One is the needs of scientists in an academic medical center are varied. We've got clinical, kind of traditional clinical trialists who are, more focused on small data sets, all the way to, data scientists who may want to, apply deep learning methods or use images. This is so many different needs, but to support all these different needs, there's so many different skills that oftentimes clinicians and other scientists don't have, that we really have to develop among our staff too. These are both technical and scientific, statistical. One thing that we found really helps accelerate time to science is by bringing together this troika of roles of clinician, biostatistician, and informatician. These types of partnerships we really saw during the pandemic in the early stages were fantastically crucial. But kind of looking at some of the data, there's trade-offs. There's always trade-offs of using things like OMOP, where we get breadth quickly, but we always kind of there's always limitations there, and kind of have to go back to depth into the source systems. That can take a bit of work. And finally, the regulatory aspects are really challenging, but investigators, I think, rely on early consultations with informatics and statistics and legal and IRB, privacy office, and others to get these sorts of things approved. So kind of bringing all this back, 3 things that research informatics can do. That's get EHR data, collect novel measures, and integrate data from disparate sources. A great deal of this is indeed this, quote unquote, janitor work, but our capacity continues to be strained, and self-service tools are really a way to help democratize access to data, so long as we have the appropriate guardrails in place. But we've learned over time that it's important to remind investigators that data are free, but thoughtful interrogation has a cost, and that's either through engagement with our group, engagement with our biostats colleagues, or just someone doing it on their own. But that really comes out of someone's time and money at some point. And through ARCH, what we really aim to do is match investigators with the right tools and services with respect to a number of different factors. I think we've had success over time doing this. It takes a village to deliver these types of services, and I'm unbelievably thankful for all of the efforts of our team. We are hiring, and we need more great staff and faculty to join us. Very thankful for the funding we received from the NIH through our CTSC, other sources, leadership we received over time, and I'm happy to answer any questions in the time that remains. Thank you.
Tom, thank you so much. That was really a great state of where we are today. I had a comment. Your janitor work of 800 ETL scripts, I might call data engineering, especially when those scripts, the data changes on us, that drifts on us. It requires monitoring and data quality and quite a bit of DevOps to actually manage correctly. So I also thought you did such a good job describing the different networks. You support them all at Cornell. And I find that was really, you serve slightly different purposes with the different networks. But I did think that really fed into a lot more complexity because you had to feed all the different data models around them. So you described how OMOP is now behind N3C and behind all of us. And then you described the ACT network and Trinetics and with I2B2 and PreCoreNet. So how do you see that changing over time? Because you're right, you need to move higher up the stack and have, and try to centralize and then have, subsets. So I guess that's my first question. How's that?
Great question. We are bringing more and more into OMOP because we've gotten pretty good at running our OMOP ETL. On a quarterly, and we're trying to get like a monthly refresh at this point, and push more stuff on top of OMOP. So when we got started with Trinetics, for example, we were providing I2B2 data. We don't do that anymore. Now we provide OMOP data. With I2B2, we kind of want to keep it around for a number of reasons, but we are likely to shift from using the I2B2 data model to using OMOP underneath with this I2B2 on OMOP set of tools that the Harvard folks have released. So more and more towards OMOP, I think, and exploring some of the related Odyssey tools like Atlas. But it's all towards this shift toward self-service a bit more, so that we can kind of free up our staff, because they still need to be involved, but we want them to be operating a little bit higher up.
I would agree. Also, Trinetics now can be fed via OMOP. And so you're right. I do feel like if we can have at least one data model that can feed the others, then you can have multiple instances. I also really like the way you describe research data repositories. Here, we call those internal clinical registries. We're collecting all the data for a clinical user group to do studies, and how you're right, you combine REDCap and other data sources that they might have about within their clinic, as well as giving them versions of their data. Have you thought about bringing to them images or other types of data along with the EMR data?
Yes, and in fact, we've had a couple of projects with our radiology colleagues where we brought those together. And just yesterday, we're on the phone discussing, okay, how can we do this at scale? So I think that's one of the next things we'll be doing over the coming months.
That's excellent. And I guess the other question I had, I mean, this was great. It really was a great state of where we are today in the field. And so I really appreciate that. I guess the last question I have for you is, you described all this work to get data, but you recognize that really that's just the starting point for people to use applications and build models, and then have those models hopefully be validated and then brought back into clinical care. So have you done work on trying to provide common analytics environments for the common tools that researchers need within Cornell?
We have something called the data core, which I think I illustrated on a slide early on, but didn't really get too much into. Today, the data core is a secure remote desktop environment, most often running Windows. Data goes in, data doesn't come out. And you can get, R and Python or whatever packages you want installed on that environment. This has been particularly helpful for studies using data from CMS, where there might be particular security and privacy requirements that we need to meet. But now we're in the process of kind of shifting that into more cloud offerings, and embracing Linux as well, so that we can support more data scientists.
Yeah, I see a couple of questions from the audience. One of the questions I like is, how do you see AI being used? I thought you presented a couple interesting models for it, for actually trying to use it for just eligibility criteria itself, for finding potential clinical trial, people that you might want to, recruit for a study.
Great question. There's a lot going on in this area, and I think some great things that are coming through the Odyssey community. And now we're starting to see, I think, a couple of commercial entrants as well, companies like Deep6 and Beck Health, that are designing tools to bolster trial enrollment, and also help rescue trials, to identify patients using structured and unstructured notes. So I think there will be a lot of activity in that area over the next few years.
Yeah, so that was great. So thank you so much for coming today and presenting. It was really great to compare notes with our initiatives. A lot of it is just language differences, what we call a registry or a precision medicine center, and our platform. But it really is great to see that we're on the same journey in trying to empower our clinicians to be able to answer questions. So thank you so much again for a wonderful Grand Rounds.
Terrific. Thank you. I really appreciate it. Take care.
Great. Thank you. And that concludes this Grand Rounds, and we hope to see you next month. Have a great month, everyone. Thank you.